This is a presentation on the feeding methods of the Stegosaurus, trilobites, mites, and theropods. Section 0103 by Mark Wong, Alex Rifkin, Matt Falconer, and Freddie Whitney.
The first essay that our group is going to discuss is about the bite mechanics of the Stegosaurus. The Stegosaurus was discovered and named in 1877 by Othniel Charles Marsh. The teeth of the Stegosaurus and its feeding habits have not been researched to a great extent, leaving much speculation on whether it ate large tree branches by standing on its hind legs and reaching the top of trees, or if it ate shrubs and bushes from the near ground. Evidence of damage to the teeth is speculated to have been a result of normal tooth-to-food contact, and the bite force has not been tested until now. Artificial computerized models, which are based on the measurements and the remains of the tax's tooth crowns and jaw structures, were created. Since the Stegosaurus, as most herbivores, did not have different types of teeth, the positioning of the teeth inside the mouth did not matter. The bite force was calculated based on skull proportions and teeth size and shape. The model was tested on recreated models of tree branches and shrubs from the time era the Stegosaurus was alive. Results indicate that the Stegosaurus had a weaker bite force than a modern-day dog. The size, shape, and the amount of teeth inside the jaw helped lower the stress that the teeth took. Jaw bones did not feel much stress from biting into smaller plants, vegetation, and shrub. When applied to thicker tree branches, a total force of 275 newtons was needed to chew. This caused failure in the teeth enamel and damaged the jawline. A much lower than expected force by the jaw was found to be sustainable for the tax to continuously chew. This analysis showed that tooth morphology is efficient in dissipating the compressive stresses generated during bite. Minimal amount of stress is transferred to the jawbones.
We now move from the Stegosaurus to the trilobite. Now for a little background information about the forked hypostome of the Isotelus-type trilobite. The Isotelus is the largest type of trilobite, and the hypostome is located under the head plate, and it's a backward-facing mouth apparatus. The Isotelus has a large, robust forked hypostome with little mobility. As we've learned in class, trilobites were a group of ancient marine arthropods that roamed the seas throughout the Cambrian period. Some functions of the hypostome have been ruled out, including the macropredatory role, filter feeding, and its use as a weapon. We know a little bit about trilobite feeding methods, such as the fact that they had a ventral food groove that passed food particles to its mouth, and that some trilobites were predators, while others were filter feeders. The big question: what was the function of the unique forked hypostome of the Isotelus-type trilobite? As you can see in Figure 1, the hypostome of the Isotelus featured two forked tines that each had wavy terrace ridges on the inner surface. The size and shape of this hypostome made it difficult to position and handle large items of food. Figure 3 shows that as size increases, the number of ridges per 2 millimeters decreases, and the ridges are actually added during growth. As can be seen in Figure 2, the ridges face the opposite direction of movement in the food group, creating a phenomenal grinding surface. By combining our analysis with analysis of modern crustaceans and arthropods, we have implied that the limbs are providing the force on the hypostome, and that these trilobites sucked up small critters with their coccyx and ground the cuticles of these prey against the hypostome. This feeding method is similar to that of the modern horseshoe crab. There is the potential for another use as a burrowing apparatus or aiding in the direction of burrowing, but that does not appear likely. We have concluded that the hypostome is immobile, and the regularly spaced ridges on it form a great grinding surface. The food is passed through the ventral groove to the mouth parts. There is potential for future research, though, as the hypostome as a burrowing agent, and also current specimens do not show any wear on the ridges.
Now we move from the troubled trilobite to the mighty mite. Charred plant fossil remains found in the Preggenhaus quarry in Germany demonstrates a plant-arthropod relationship that is rarely preserved. The scientists discovered a nymph-orbited mite on the pollen sac of a mid-Cretaceous angiosperm. Now for the big question. Are Mid-Cretaceous mites pollinators or decomposers, and why was this mite on the flower? Scientists observed the fossil for clues. In order to get the answers, the scientists cleaned off the fossil. Then they looked at the flower under a microscope, and while observing the remains, they stumbled on something interesting. Results showed that the mite had its mouth on the plant's pollen sac, which suggests that it was eating the pollen. They also observed the presence of a fungus, a known decomposer. This tells us that the plant was dead on the mite's arrival. They also discovered the presence of fecal pellets, the orange-red dots in the picture below. This showed the mite had been there for quite some time. This evidence shows us that the mite was a decomposer in its environment. And now for our last look at mites. Mites back in the Mid-Cretaceous are like mites today. They are both decomposers in their environments. This fossil find also shows us a rarely preserved interaction between plants and terrestrial arthropods, as well as help us build the decomposer food chain.
Our final paper is on theropod feeding strategies. This study covers all types of carnivorous theropods. However, there is little evidence that theropods consumed bones. In addition, juveniles are also very rare in the fossil record, which might be because of theropods targeting them. The big questions are, did theropods preferentially hunt and consume juvenile dinosaurs, and did theropods purposely eat bones? In the study, the scientists examined the fossil record in extreme depth. They looked at bones with bite marks, and they compared theropod hunting strategies to those of modern predators. In addition, they unfortunately could not examine the theropod coprolites because they are very rare. They also examined stomach contents. They found that theropods hunting juveniles agrees with the evidence, and they determined that theropods did not purposely consume bones. The absence of juveniles is likely due to theropods targeting them. In addition, the consumption of juveniles might have provided theropods the necessary nutrients that they needed. In conclusion, theropods most likely hunted like modern predators and tried to eat whatever was available or the easiest. This is only a supported hypothesis, and theropods could have hunted differently.
So to summarize, trilobite hypostome was used as a grounding surface to break the cuticle of small prey. Carnivorous theropods did not intentionally consume bone, and they targeted juveniles and smaller prey. Stegosaurus' bite force was incredibly inefficient. Mites are important decomposers that enjoy eating pollen.